of the Gil Omen Computational Proteomics Award of the US Human Proteome Organization, and he's also the recipient of the Beeman Award of the American Society of Mass Spectrometry. These are very distinguished recognitions, and we are very happy to have Nuno joining us today and sharing the work that his team is doing on MassIVE. Thank you so much, Nuno.
Thanks, Olga, for the very nice introduction. Let me share my screen so we can get started here. I still need to do that myself, right? There we go.
That's right.
All right. Thank you for, again, for the very nice introduction. It's a pleasure to be here today and to get to share a little bit of the things that we're doing at MassIVE, and in particular with MassIVE Quant, to enable public proteomics data sharing and reuse, because that's really one of the main things we focus at MassIVE. And it's also a pleasure to be here today with those of you who stuck it through and you're still here. This is the core group that really is most interested in things, and that is the perfect audience. So,
and Nuno, sorry to interrupt. I forgot to mention the very important thing. Please use the Q&A button for questions to the speakers. We will answer the questions in writing as possible, and if not, I will be moderating and interrupting the speakers if some questions need to be voiced out. Please don't hesitate. We hope that this discussion will be interactive.
Perfect. And this was not scripted, but it was the perfect moment to say that, because I was just about to say that it's going to be more of an overview of the things that we make available at MassIVE. And then we want to, if there are questions about it, if you want us to elaborate on it, please go ahead and let us know. I'm not sure how to interpret that question, so if you could please elaborate a little bit.
I will.
Okay, thank you. All right, so let me go ahead and get started here. So you've all hopefully at this point heard about Proteomics Change, the global consortium for sharing of mass spectrometry data. And there are several repositories that support this effort in different continents. And to me, one of the most impressive things about this effort is just how much adoption we have seen over the years for the sharing of public mass spectrometry data. So if you look back in 2016, just 6 years ago, there was just 4,500 data sets that were available at that time. And today we're already at over 20,000 data sets. So this is a really very significant growth, 5 times more data pretty much. And you can see that the pace of growth keeps increasing. The first 3 years, it just doubled, but now it's doubling again. So if it keeps going at this pace, you can see just how quickly data becomes available. So then the question that we thought about at MassIVE, as a repository, we currently have 17,000 data sets, almost hundreds of terabytes of data, billions of spectra, and there is this general idea of thinking of a repository as just a place where data goes in. And for many users, this is kind of a view of the repository that they hold, which is at the end of the project, when the paper needs to get published, data just gets pushed somewhere into a repository, and that's the end of the story. We see it differently. From our perspective, we want the data to be useful. So it's worrisome for us when we look at what actually gets deposited. You see that a lot of data sets are deposited. There's a lot of raw data that is deposited. However, the identifications that are attached with the data set correspond only to a very small portion of what is uploaded. And for the vast majority of cases, quant data is not there at all. So, or at least it's not there in a format that you can reuse it. So if things are bad in terms of identification results at about 6%, it's even worse in terms of quantitative data to be shared with those data sets that are available. So because of this, the way that we developed MassIVE was to be not just a repository where data goes in, but a place where data can be exchanged between repositories, and data sets that initially get deposited elsewhere can be imported into MassIVE and regularly are. The data can be converted to open formats. If you want to look at any of the available data sets, you don't have to have the software for the instrument where the data was generated. The data is reanalyzed in the system to add value to the data, to add new identifications, to add new quantification information, and we'll talk about that in a little bit. And this is done with a number of workflows that are available for discovering modifications, running proteogenomics analysis, doing quant analysis, and then visualizing those results online. So to give you the ability to interact with the data, and especially these large volumes of data, without having to download too much, or hopefully in some cases even anything, into your computer until you know that you're going to need it. Of course, you're always welcome to do it if you want to, but you wouldn't have to. So let me talk a little bit about the different kinds of things that we have available for this purpose. So when you go to MassIVE and you have the splash page for the repository, this is what you'll see. You have some statistics on what's available at this point, and you have the standard two functions of a repository, which is you can access all the public data that's available, or you can submit new data sets. And this is also the route to go and do other kinds of analysis in data sets, as we'll discuss in a little bit, the sorts of things that you can do. So, but this is, for us, the starting point. It's how data gets in and how you can access data and get data out. So when you look at a data set page, it generally looks something like this. And I'm going to highlight a couple of things that you can do on data set pages. So the first thing is, if you see the proteomics change identifier for a data set, this is the MassIVE identifier for a data set. For proteomics data sets, you usually have both. But of course, for metabolomics and small molecules data sets, which we also have for the GNPS repository that Olga mentioned briefly, they're not a part of proteomics change because it's metabolomics data. So in that case, you just have the MassIVE identifiers. If the data was imported from elsewhere, then we have links back to where the data came from. So we keep the full provenance of the data. And in this case, it's CPTAC data sets, which they're pretty much imported into MassIVE, because we can then do both showing the results of the original analysis of the data that's deposited at CPTAC, but also give the ability to reanalyze the data and get information about that data. Now, if you are the owner of a data set, you have the ability to change the data set over time. You can never delete things from a data set, but you can add files. It often happens that some files were forgotten in the process of building the data set. And during the review process or later, you find out that it's important to add a few files to a data set. You can do that automatically through this option. You can add or update metadata. Sometimes the contact person for a data set changes, or in particular, you can change and add publications to a data set, which over time, that's likely to happen since you submit the data set before you publish the paper for the reviewers to be able to access the data, but then you can come back and add that paper afterwards. Now, while the bottom row is actions that are only available for the owner of the data set, the top row is available for everyone. And as I mentioned briefly earlier, you can convert the spectra in a data set into open formats so that then you can use those for reanalysis with open source software tools. You can actually reanalyze spectra directly at MassIVE without having to download them. So for this data set, as you see here, there's the 19 million spectra in there, close to 20 million. Of course, you can download it, but as you see, the data set size is over a terabyte. So, that can be a deterrent to reanalyzing the data on your own compute infrastructure. So that's one situation where it's more convenient and simple to just use it here. And there's a number of workflows. There's the MSGF workflow that's used for database search. There's a number of other workflows, spectral libraries, proteogenomics, blind modification detection, so on and so forth. And especially relevant for today, there's a MaxQuant workflow that you can use to run MaxQuant directly in the system. And there's an MSStats workflow, which also enables you to run MSStats directly in at MassIVE without having to run it locally for public data. And we'll talk a little bit more about that in this morning's session. Once you run a reanalysis, and you can do this locally or at MassIVE, you can add that reanalysis to a data set. And what that means is that it makes your work of reanalyzing the data set visible to the whole world, such that when anyone goes to the data set page, they also get to see the reanalyses that were done and attached to those data sets. And you get your own identifier that you can also use in your papers to guide people towards the results of the reanalyses that you did and shared with others. So once these reanalyses go in, what can you do with it? It's like a data set. You can share it, you can make it available to others, people can download it, but you can also look at them online. So there's a question here, is MaxQuant on the system here? Yes, we run the Linux version of MaxQuant at MassIVE, and we regularly use it for label-free data sets, and we have quite a number of those results already attached in MassIVE Quant. So yes, it is available to run it here. So once you have these reanalyses, one important thing that you can do, again, you could download them and then do whatever you want to do offline. We definitely support that and encourage that. But if you want, you can also run these comparisons directly on the system. So there's a workflow that you can use to compare results of different reanalyses. And what you do is you can select the jobs that you have in your account, or you can select from public data. You can select from existing data sets or from existing reanalysis and choose to compare those jobs. When you do that, you get a result that compares at the spectrum level, peptide level, protein level. And because in some cases people use different databases where the same proteins have different identifiers, you can look at remapped proteins, in which case it just takes the same peptides and makes sure you have the same protein identifiers for both comparisons, and gets you to see, you know, how do the original data set results compare to the reanalysis results for that data set. And here's an example where the original data set only considered the basic sample handling modifications, but if considering open modification search for this data set in particular, there was a lot more that could be found, and that it would have been pretty much missed in the original analysis. So the ability to reanalyze data with a different set of algorithms can really enrich the things that we can know about these data sets. So let me now then talk a bit more about the ways in which you can explore the results that are available at MassIVE. So in particular, all of these many reanalyses that were done at MassIVE, and we've reanalyzed tens of terabytes of data, you can go to this MassIVE search option that's right below the access to public data sets, where you see the list of data sets. Here you can see the list of all identifications. You can come in and search for peptide, modification, search for protein, and it tells you all the data sets or all the proteins identified or all the modified variants of a peptide that have been identified. If you're interested in a modification, it tells you all the peptides where the modification were detected. And right now, this system contains close to 1 billion PSMs, or peptide spectrum matches, that have been accumulated over all of these data sets that are publicly available at MassIVE. Now, if you're not so much interested in the data set results directly, but you want to reuse the information that is derived from those data sets, one way in which MassIVE helps with that is by constructing spectral libraries from these data sets. So right now, what we have on the system is the first version of the MassIVE knowledge base, or KB, spectral library. And in this case, it had about 2 million precursors from over 30 terabytes of proteomics data. But now the latest version of MassIVE KB, which we'll be releasing in a couple of weeks at ASMS, now contains over 6 million precursors for pretty much all human genes. This includes some synthetic peptides, because some of these proteins have not yet been observed in vivo, but it's added to the library to give you the most possible coverage of the whole human proteome. So it triples the previous size, and it's by far, I believe, the largest library that you can have access to for this. Question here, is there any quality control of data uploaded to MassIVE? That's a good question, because quality control is a different thing to define in the sense that experiments can change what you expect to see in the data. So it's important that the question about quality control be asked carefully. But one way in which we address this is the following. We have a workflow that we can use to assess the mass errors and the modifications and to learn about a data set. And once the results of that workflow are typically used with the search workflows to then go and see how much we can extract from the data sets. So in a way, an indirect way of doing this quality control is that we have the search results for a data set, and if they don't look good, it does tell us that either we're missing something about what's in the data set, or there's an issue with the quality of that data set. So we have another question here. Do new or current contributors ever have issues with the data being reanalyzed and the results published, specifically peptide proteins and PTMs? Okay, so it's a good question. Everything that is done in terms of reanalysis is only for public data, of course. We never touch any private data at any time. And number two, when a data set is submitted, there is a license for a data set. And by default, the license is open access, that anyone can use the data for whatever purposes, including for reanalyses such as this. But there is an option to select a different license. However, over 98% of data sets are submitted under the open access license. So there are no concerns there in terms of the data being reused. We never, if necessary, we could go and inspect the licenses for the other data sets, but up to date, we actually only needed to use the data sets that are completely open access. So we have had no concerns with that. But it is an important thing to keep track of.
I think the question was also about whether people get upset that the new reanalysis results differ from the original published results.
If they do, it's in private, because I haven't heard anything yet. No one's complained to me, but I'd be glad to address any concerns if there are any, but not today. Is this only for the human proteome? At this point, yes, but we have started to build spectral libraries for other species. We have a different effort called Proteocarta, where we are building those spectral libraries and making them available. So please feel free to contact us if you're interested in those, and I'll be very happy to share the link to that resource also. But this MassIVE KB here is currently free. It is for humans. All right, so if you want to explore what information is available in a repository for a protein, and you want to have a better sense for protein coverage, like you want to see which peptides are usually detected for a protein because you're designing a targeted experiment, or you want to see if the peptide covers a splice junction or maps to multiple proteins, that kind of information is available in a Protein Explorer resource at MassIVE, where everything is integrated at a protein level interface instead of, here it's a spectral library. MassIVE search is spectrum level. So the final thing I wanted to mention before diving into the quant part, which I really want to, is one of the main things that our colleagues in genomics have had for a long time is the ability to blast sequences, that they come with a new sequence they acquired in an experiment, and they can blast it against all the data that's available in a public domain. So what we've done recently at MassIVE is to develop a way that you can do essentially MAST, which is the mass spectrometry equivalent of BLAST, where you can bring a spectrum and essentially blast it against all the spectra in the repository. The first version of this system was published in Nature Biotech for metabolomics spectra at GNPS. But what we've done now is we've expanded this service and we've made it much, much faster. Back in the day, you had to launch a workflow to run this search, and this could take anywhere from 5 to 10 minutes. So it's not like you have to wait overnight, but you'd go for a coffee. You don't sit in front of your computer waiting for those results. However, this new updated service is now much, much faster. And the way that you use it is you use a universal spectrum identifier. And there's also a paper in Nature Methods describing how this works. But basically, it's very easy to construct one. You just say mzspec : you put a data set identifier, raw file name, scan number, and there you go. That's all you need to identify a spectrum across the whole set of public data. If you want to put an ID on it, you can, but you don't have to. And then what it does is it searches across the whole repository, either for spectra that are exact matches or for spectra that are off by a certain delta mass. So it also allows for analog matches. So there's two kinds of searches, and you can search in two contexts. You can search only the spectral libraries. So if you want to only search against reference spectra, search the libraries. If you want to search the whole repository, you can. So it takes a little bit longer. If you do analog search at the repository scale, it takes under a minute. So it's still relatively interactive. But if you're doing exact matches or you're searching libraries, then it's typically under a second, unless there's some load in a system. And then you can see across the whole repository, how often do you get spectra that have a delta mass or differ from your spectrum by these many daltons. You can see the list of data sets where those are found. In here, it's shown by delta mass. And you can see whether the spectra are identified in those data sets or not. And of course, there's a list of individual results if you want to follow up on specific sets of results. So it's another way in which we enable accessing data at the repository scale. I'll be happy to share the citations afterwards if there's a question about which one specifically. I don't know, nature protocols. I'll be glad to share that afterwards. So now I want to make sure we do come to MassIVE Quant, because this is something that was a very interesting project for us, that we really wanted to address these limitations, that to prioritize anything in mass spectrometry, to be able to do any biology with it, you need quant information. I know I'm preaching to the choir here, but this is, as you also well know, a complicated endeavor, and we have this whole May Institute on that. And people do things differently. They use different tools. They have different workflows. They combine tools in different ways. And to be able to support all of this, there really was a need for some way to share this information in a way that could be enabling others to understand what was done in each analysis and reproduce it, hopefully, so that then you can change things and do things differently if you'd like to. So MassIVE Quant was put together to address this need, to try to meet this need by allowing us to submit reanalysis, to submit data sets, to submit original results in the way that they were analyzed. So rather than have to force too much in terms of you have to write new files, you have to define new formats, you have to do all of that, we tried to meet researchers where they were, so that you could take the results files and the MStats files that you already constructed for your analysis and use that for sharing your analysis with the world. So that's how MassIVE Quant was originally defined, together with Mina and Olga. And at UCSD, the group that put it together was Ben, Julie, and Jeremy, who still continue to work with this and help us build it up. So let me take a quick overview of some of these major features that we support at MassIVE Quant. So you can share the metadata for a data set. Obviously, everything starts with that. If you don't know where to find the proteomics data for each sample, then everything else is, of course, not possible to do. So at MassIVE Quant, we originally built the system based on the MStats input format. So that particular file that you use as input to MStats, where you define for this raw file, this TMT track, is this sample, this bioreplicate, so on and so forth. That is an input that can be processed directly at MassIVE Quant. But we also support the new format that has been recently defined, and Yaset probably is going to tell us a bit more about this in this morning session, which is now a standard format that we can use across all of proteomics change and is not really tied into any particular tool for defining this metadata related to samples. And these use controlled vocabularies, which make it easier to, not refer to the same thing with different terms. Now, in addition to supporting metadata, it was very important for us at MassIVE Quant to also support the sharing of quantification results and the sharing of the statistic analysis of the quantified analytes. And I say quantified analytes, even though the vast majority of cases are doing protein analysis, but it could be peptides, and it could be small molecules, and it could be modified peptides, could be other things. But the important part is we want the data sets to have, of course, the raw data, the ID results, the metadata, but also the results of the quantitative analysis, because if we don't, we're still stopping short of fully sharing the quantitative analysis of the data set and enabling others to check what happened with those analyses. In addition to the quantitative data set formats, we also provide executable workflows at MassIVE for the quantitative analysis of data sets. And in particular, I already mentioned briefly a little bit ago, we have the MaxQuant workflow that is available in the system. It says still here pre-production. I have to go and check if we already deployed it or not. It's certainly been ready for deployment for a while. We tested it very exhaustively. But if it's not there yet, it would be only because the documentation needs updating. The workflow is definitely ready. But we definitely have had in production already for a while the MSStats workflow, which allows you to run the MSStats analysis of quantitative data that is available in the system. And all of these, I just want to mention here quickly, they work for public data, of course, but they also work for private data. So at MassIVE, we allow you to have a private space for your own research data. And that's actually where you first put your data before you make a data set. But you can also use it to analyze data at MassIVE for the research purposes. We also have the MSGF database search, which has support for exporting isotopic labeling quant information, which you can then input directly into MSStats. So let me give you a quick example, actually, of one reanalysis that's currently deployed at MassIVE Quant, that's publicly shared at MassIVE Quant, and it was done in the system using these workflows that I describe here. So MSGF was used for database search. This is a TMT data set. And as a result, when the job concludes, you have these options at the end in the job status page that includes MSStats inputs for all the proteins, including reference proteome, isoforms, etc. Or you can export protein mappings only to canonical proteins. In some cases, if you're interested more in a gene level analysis, that can make sense. If you're interested in isoform analysis, then you can take the full set. Both are made available as a result of the database search. Then you can run MSStats in the system, and you can select your conditions. You can define if you do all versus all. You can define which pairs of conditions you want to compare to each other. And then you have interactive results views that allow you to explore the results. You know those tables that you get with all the comparisons, p-values, and log-fold variation. All of those are available in there. And then once those results are ready, you can go and attach those data sets to, you can attach the reanalysis to the data sets if you want. You don't have to. You can keep it for your own research purposes and leave it at that. But if you feel like it's interesting to share, you're very welcome to go and do so. So question here on the workflows, how are they created, how are they reviewed, and who does the review? So the workflows are created in our system using an XML based format, and they're all open source. We make the workflows available on GitHub, so anyone can go and see what the workflows do and how they do it. And I also want to mention that every time you attach the results to a data set, there's a link back to the actual job that was used to get those results. So it tells you which version of the workflow was used to get those results, it tells you which parameters were used to get those results. So you have all this complete information to completely reproduce it, or if you find any problems with it, you're very welcome to point them out. And it happened in the past. We had to correct some of the jobs because we found out there was a wrong parameter, or there might have been a bug in the code somewhere. So this is very important for us, so we make it all available so anyone can see it. Now, I do want to mention another principle that we apply across all of MassIVE, both at MassIVE GNPS and all of these resources, and that is, number one, doing the best we can at our level in terms of we test the workflows thoroughly, we use them ourselves, so in a way we eat our own dog food, and that really makes it go through a lot of cycles of testing. Number two, we're 100% open. We make the workflows available, we make the searches available so that others can also look at it. And three, we want to be responsive to any issues that come up. So if anyone finds bugs or results that don't make sense or incorrect search parameters, we're very glad to go and recheck that. This is in contrast with, some sort of sense that there's a higher authority that could determine by decree something is right or something is wrong. We just prefer openness, and we make it available to everyone. And if any issues are detected, we go back and try to fix them. How do we do the quant at the peptide level? So this is more something that is done at the MS-Stats level. What we do is the way to do this in MS-Stats, I don't know if this was covered at any point during the course, but what we do is essentially each peptide is reported as its own protein, and then MS-Stats does what it usually does. Now, of course, you're not going to have the same kind of quality of quantity, and this is going to affect the stability of the analysis, because when you have a protein with many peptides, of course, you're going to get effects on some peptides that are not in others, and the protein level kind of averages that out, and you can get a better analysis at the protein level. At the peptide level, you have to take it a bit with a grain of salt, because there's going to be more variability, because there's less ways to average out, spurious effects happening on specific peptides. But it is possible to do it, and it is available as a way to do the analysis here. So just quickly, here's a brief look at what these interactive results views look like. And you see here, like the table that you get as the MS-Stats results, you can filter for different proteins, you can sort by full change or p-value. If you click on the viewing icon for a comparison, you get to see how the protein abundances were detected across the different conditions. And you get to see for the different pairwise comparisons between conditions, what the log fold changes were. So all of this information is available there. You just click the button, it opens up, and it shows these results right there in an interactive manner. Now, one thing that I want to mention that is a very recent development that we're actually releasing this month is the Protein Explorer extension into the quantitative world. And if you go to the MassIVE Quant page, there's a number of things you can do. You can browse what's submitted in terms of data sets and reanalysis. You can submit your reanalysis or access reanalyses. We'll hear a bit more about that in a little bit. You can run MS-Stats, or you can pair quantitative results. You can do all of those things. But I wanted to mention this new feature that we just put together that we found meets a need, that whenever we see a new protein being compared and getting some differential abundance in a certain p-value, how commonly is this protein found to have a certain differential abundance or being so significantly differentially expressed between conditions? And what this new interface allows you to do is exactly that, is to give you a protein level entry point into all this quantitative information that is available at MassIVE Quant. So this UI, if you just look at it at a high level, when you open the MassIVE Quant Protein Explorer, you essentially have three panels. On the top left, it shows you all the proteins that are available in MassIVE Quant, and there's close to 90,000, part of it because of isoforms, part of it because of multiple species, and honestly, a little bit of it because of different protein identifiers being used in different data sets. But there's plenty of events, meaning comparisons in MSStats analyses. And then some of those comparisons are significant. And we see here for these different proteins, the number of events that there are for them and how many of those comparisons were significant. Top right is a volcano plot for any protein that you select in here. You get to see all of its events or comparisons at the repository scale. So here we're seeing for histone H4, all the 700, and actually, no, not all 794. We're seeing the 139 that are significant because there's the p-value cutoff of 0.05 here, and we're not seeing all of those under it. And if you want the details for each of these dots, you can get that in a table that shows here at the bottom of the page. So in a bit more detail, as I mentioned, this protein selector table, you can filter for your protein of interest, or you can sort or filter by number of events, or you can sort or filter by the number of significant events. If a protein has been detected as significant many times, but you're most likely to go and look for your protein of interest and then see how many times was it analyzed and how many times was it differentially abundant. Then when you get to the volcano plot, what it does is each of these dots, it shows you what the comparison results were in one reanalysis or in one data set. So we see here, for example, this protein, histone H4, in this data set had a log2 fold change of over 100-fold, and it was an extremely small p-value. And you have the overall distribution across all the data sets. Of course, most of the observations, it was not significantly differentially abundant. Question here, chance to integrate other tools such as FactPipe or on MassIVE. So that is actually, we do allow you to import search results from other tools into MassIVE Quant. So as long as they write their results in an open standard format, such as MZ-IdentML or MZ-Tab, those can be imported into here too, and then be used for other analyses such as MSStats or other tools that take as input search results. So the volcano plot gives you the ability to visualize all of these comparison events at the repository scale. And then if you look at the details, you can then find, of course, what data set the comparison was done, which reanalysis the comparison is reported in. You can see the names of the conditions that were compared, log2 fold change, adjusted p-value, and the file that contains those results. So all of this information is available here for both filtering and visualization information. Right. Which is this point here, you can filter or sort by any of these fields. So very quickly, let's say that you wanted to inspect these SAA1 or 2 proteins, which were claimed markers of COVID infection. This has been reported in a couple of papers, including some of the high impact papers that came up early in the pandemic. And so the simple way to do this is we would go here to the protein box and you would type some part of the protein identifier. I just typed vertical bar SAA because I want to see all the SAA genes. And a few come up, both human and mouse. And you can see how many events for each of them and how many of those were significant. Actually, the way to make this show up is you click the Apply Filters button. So first you type this in here, you click the Apply Filters button, and that filters this table to show only the proteins that contain that substring. Then you can sort by the number of events. And in this case, SAA1 has the most differential abundant events that were significant. And if you click on the number of events, that updates the table on the bottom and the volcano plot on the right. So yeah, this is what it does. You click over here and it gives you the details down here. And then on a volcano plot, it gives you the ability to see all of the different events. And if you hover your mouse over a point, it actually tells you the actual values. And we see here for this particular differential expression, it was a log2 fold change of 2.7 with a p-value, with a log10 p-value of 10, so very significant detection. And it's the one that we see down here. So if we click on it, it then filters the table and you get to see the details for that particular point of interest. So in summary, I know we covered a lot of ground. I wanted to give everyone an overview of the sorts of things that we do, and I'm glad to answer any other questions you might have about the details of how to use it or how to apply it in your particular searches. But as you can see, there's a variety of different ways in which you can explore and interact with massive spectra, massive search results, and massive data sets, including ways to reanalyze the data sets online if you'd like. And that includes massive quant, which allows you to do these things for quantitative analysis workflows. We take this very seriously. All of the identification results that we get only mean something if we can put it in a quantitative context to have some biological meaning. So these workflows are used very frequently and updated very frequently also. And now you can explore all of these results at the repository scale. Just wanted to give a quick note here. The kinds of data that are supported. Massive Quant supports both data-dependent acquisition or data-independent acquisition, label-free or labeled, targeted experiments. Anything you can input into MSStats is something that we can parse and share results for in Massive Quant. There's a question here for the online versions of software packages, such as MSStats or MaxQuant. Are they fully featured or simplified in any way? They are simplified in some way. So they are designed for the most common kinds of searches that people want to run. But there's plenty of things you can configure in those input forms. And we found that for all the public data we reanalyzed, all the parameters necessary are available in the input forms, but we're always opening to listening to comments that others may have of things that are missing that they can show to us why they're important. So we'd be glad to hear from you guys if you feel like anything else should be added in there. So with that, I just want to wrap up by acknowledging the people who actually did the work of making all of this in the system. Julie's done a lot of work for the recent version of the MSStats input form for SDRF. Ben was the person who actually helped put together the first version of the MSStats workflow in Massive Quant. And Jeremy has been instrumental now with the whole Massive Quant infrastructure, as well as with the Protein Explorer interface we put in place recently. And of course, Mina and Olga, without whom none of this would exist. They were great in helping put together the system to begin with, helping us design it, and leading the effort for bringing it to where it is today. And with Peter and Ming, where we've prototyped many of these recent developments at Massive in the context of small molecules for GNPS, and where the first version of this masked service was also initially implemented. So one other question in here, is there support of N15 labeling as used in plant sciences? It's certainly used in a number of places. I would maybe bounce that question back to you, Olga. Is this something that MSStats can take in as input?
We would need to look at the format of the data. I cannot tell, maybe. So maybe an answer to this question is, please reach out to us, maybe give us an example of a data set and we can comment on how things can work. But I think from what Nuno is saying, if it can work in MSStats, it can work elsewhere. Is that the right answer?
Right. If it works in MSStats, it can be used as an input. Absolutely.
Okay. Well, thank you so very much, Nuno, for the introduction. It is very, very interesting. So